transamination and oxidative deamination. Today we discuss these two processes in the cells. First, from these two, we discuss transamination. Transamination is a process of transfer of an amino group from an amino acid to a keto acid, which results in the formation of new amino acid and a new keto acid. So this process is called transamination. Simply we used one word, transfer of amino group from amino acid to keto acid. Amino acid to keto acid is called transamination. The transamination catalyzed by transaminase or aminotransferase enzyme. For this process, coenzyme is also needed, that is pyridoxyl phosphate. See in this diagram, it is one amino acid, alpha amino acid. That alpha amino acid containing amino group, that amino group transferred to keto group. See, here it is the keto group, C double bond O containing group, keto acid. Amino acid containing amino group, it transferred to keto group. It transferred to keto group. After transamination, the amino acid convert into keto acid. Keto acid convert into amino acid. Means after transamination, new keto acid, new amino acid is formed. For this process, aminotransaminas or transaminase enzyme is involved. So it is the general formula about the transamination. See, it is we discussed the transamination is two types of transamination in this. There are two active transaminases in the tissues catalyzing the transamination. They are glutamate oxaloacetate transaminase. Glutamate oxaloacetate transaminase. Simply we called GOT. Second one is glutamate pyruvate transaminase. Simply we called GPT. These two reactions belongs to transaminases. So first we discuss glutamate oxaloacetate transaminase. Here it is the best example for the alpha glutamate oxaloacetate transaminase. See, the first amino acid is aspartic acid. The aspartic acid containing amino group. And the second keto group is alpha ketoglutaric acid. Alpha ketoglutaric acid. From aspartic acid amino acid, amino group transferred to keto group. From aspartic acid, amino group transferred to alpha ketoglutaric acid in presence of aspartate glutamate aminotransferase to form oxaloacetic acid and glutamic acid. After transamination, aspartic acid convert into oxaloacetic acid. After transamination, alpha ketoglutaric acid convert into glutamic acid. These are the newly synthesized keto acids and amino acids. It is the reversible reaction also from these two. From these two means from the oxaloacetic acid and glutamic acid in presence of glutamate oxaloacetate transaminase. Amino group transfer from glutamic acid to oxaloacetic acid. Oxaloacetic acid convert into aspartic acid. Glutamic acid convert into alpha ketoglutaric acid. Means it is the forwardable reactions, reversible reaction. This is the first transamination process. The process is called glutamate oxaloacetate transaminase. The second one is glutamate pyruvate transaminase, simply called GPT. See, here it is one amino acid is alanine. Alanine containing amino group, and the keto group is alpha ketoglutaric acid. See, transamination means amino group transfer from amino acid to keto group, keto acid. So here it is alanine containing amino acid. That amino acid transferred to keto acid, alpha ketoglutaric acid, with the help of alanine glutamate amino transferase. After transaminase reaction, alanine convert into pyruvic acid, alpha ketoglutaric acid convert into glutamic acid. So it is newly synthesized products. Pyruvic acid is newly synthesized after transamination. It is one type of keto acid, and glutamic acid is also newly synthesized amino acid. These two amino acid and keto acids are in reversible reactions also. They are involved in the reversible reactions with the help of glutamate pyruvate transaminase. Series glutamic acid containing amino acid is transferred to keto acid, transferred to pyruvic acid. After transamination, glutamic acid convert into alpha ketoglutaric acid, and pyruvic acid convert into alanine. These are the two methods of the transaminase process. And transdeamination. Transdeamination is the combination process of transamination and deamination. Transamination means the process of amino group transfer from one amino acid to keto acid is called transamination. After transamination, continuation reaction is deamination. Elimination of amino group from the amino acid is called deamination. So here it is alpha glutamic acid, L-glutamic acid. L-glutamic acid is one type of amino acid. That amino acid containing amino group transferred to keto group. Keto group is called pyruvic acid. After transamination, the pyruvic acid convert into alanine, and glutamic acid convert into alpha ketoglutaric acid. So here it is, transamination process will over. From glutamic acid, amino group transferred to pyruvic acid. After transamination, pyruvic acid convert into alanine, glutamic acid convert into alpha ketoglutaric acid. See, alanine containing amino group. Here it is, the next process is deamination. The deamination done with a specific enzyme, that is amino acid oxidase. Amino acid oxidase. See, alanine containing amino group. That amino group is eliminated from the alanine amino acid with the help of amino acid oxidase to form pyruvic acid. Pyruvic acid is the keto group, and from that alanine, amino group is eliminated. Amino group is eliminated. This process is called deamination. So in this process, transamination will occur. Then continuation after transamination, deamination also will occur. So these two processes are combinedly called transdeamination. Transdeamination. And next, decarboxylation. Decarboxylation means elimination or removal of carbon dioxide. Removal of carbon dioxide from amino acid. From amino acid. So from this removal of carbon dioxide from amino acid, carboxylase enzyme is needed. Carboxylase enzyme is needed for the decarboxylation process. So here it is amino acid. Amino acid containing C double bond OH, COOH, DCOOH, carboxylic group. That amino acid containing amine group, carboxylic group. From this amino acid, carbon dioxide is eliminated. Carbon dioxide is eliminated in presence of amino acid decarboxylase. So here it is C double O. CO2 is eliminated. Finally, to form amine. Amine is formed. In this process, from amino acid, carbon dioxide is eliminated, or removal of carbon dioxide from amino acid in presence of amino acid decarboxylase enzyme. It is the decarboxylation process in cells or in amino acids. The next one is oxidative deamination. Oxidative deamination. See, here it is. Oxidative deamination means from amino acid, amino group is eliminated. Simply we called it is deamination, but the deamination process containing two major steps, two major reactions. In first reaction, the amino group of amino acid involved in the dehydrogenation reaction. It is the first step. Dehydrogenation means from that amino acid, hydrogen atoms are eliminated. Hydrogen atoms are eliminated with the help of dehydrogenase enzyme to form one intermediate product. To form one intermediate product, that intermediate product is called amino acid. Amino acid. Intermediate product is called amino acid. Here it is amino acid again participated in hydrolysis. Amino group of amino acid participated in hydrolysis to form keto acid, and ammonia group is eliminated from amino acid. So here it is simply called deamination, but the deamination process done with these two steps. One is dehydrogenation, second one is hydrolysis. Dehydrogenation is elimination of hydrogen from the amino acid to form amino acid. After formation of amino acid, that amino acid form involved in the hydrolysis reaction to form keto acid and elimination or removal of amino acid from that amino group. It is the general formula about the oxidative deamination. See, so oxidative deamination done with two steps. One here it is glutamic acid. Example we taken here, it is glutamic acid. The glutamic acid containing amino group. That amino group is involved in the dehydrogenation process. In this dehydrogenation process, NAD electron receivers, electron acceptors are involved to form alpha aminoglutamic acid or alpha aminoglutaric acid. In this first step, one NADH plus H plus is formed. It involved in the electron transport system and formation of ATP, that is oxidative phosphorylation. See, in this first step of oxidative deamination, alpha aminoglutaric acid is formed. So why we called alpha here? It is alpha carbon containing double bond with ammonia, double bond with amino group. Then that alpha amino group is involved in the next reaction of the oxidative deamination. That process is called hydrolysis. Alpha aminoglutaric acid involved in the hydrolysis reaction to form alpha ketoglutaric acid and one ammonia group. See, ammonia group is eliminated from the glutamic acid with the dehydrogenation and hydrolysis process. This process is completely we called oxidative deamination. Oxidative deamination. In this example, we take here it is glutamic acid, oxidative deamination. So here it is dehydrogenase means removal of hydrogens. Removal of hydrogen is called oxidation process. Removal of hydrogen is called oxidation process. So here it is oxidation process start with the dehydrogenation and finally hydrolysis. One ammonium group is eliminated from the amino acid.